Okay, hello everyone. I'm really excited that I'm here. My father passed away on Saturday, and I had to leave to join a family collective grieving that we have and tradition for 7 days. As you can imagine, I'm not in my office, so my internet connection might be not optimum, and if I would not be able to present everything, but I wanted to at least make sure that I will show my level of commitment that I have to this thing that we are doing together, and I hope that that would be something that would make a meaningful contribution to the field. So I'm really excited that Anna and I we have started to work together on bringing the first sort of task-based fMRI collective effort to be able to make a mega-analysis in ENIGMA addiction. And we are going to just give you an idea about why we are interested to fMRI drug reactivity and what we are going to do in just 10 minutes. And I'm going to give a short presentation for this session, and Anna is going to present for the next session. As you can imagine, Anna would be definitely presenting in a higher quality. So if you want to join the next session, you can enjoy having her presentation. So just giving you an idea about what we have. If you just take a look inside clinicaltrial.gov and look for neuroimaging outcome measures. So how many trials we already have on clinicaltrials.gov that have an outcome measure, one of the registered outcome measures that would have neuroimaging inside. What is your, I'm talking about addiction clinical trials that have neuroimaging as an outcome measure. What is the estimate that you have? Just give a rough estimate about the numbers. I'm just looking into the chat space, how many guesses we have here? 2. Okay, what are the 200? Okay, so we are talking about 200 clinical trials, 15 clinical trials at neuroimaging as an outcome measure in clinical trial. Okay, so we have 409 registered clinical trials that they have neuroimaging as an outcome measure inside. Different modalities of neuroimaging. As you can see here, they are distributed across time, with kind of increase over time, and different drugs. And then if I just show you how they are being distributed in terms of the scale, modality, and paradigm and task category, as you can see here, we have 688 outcome measures in these 409 trials. And 400 of them are fMRI. And then 342 of them are basically task-based neuroimaging modalities. And Reactivity is the most frequent task modality that people tried in as an outcome measure in an addiction neuroimaging trial. So you can see that this is basically the most frequent task-based category that people have used as an outcome measure in a clinical trial. And then when we talk about fMRI drug reactivity, we have showed in different studies, and others have done the same, in terms of if we do the same task in different clinical population, different replication samples, we can have a sort of robust response, and we have a reasonable level of replication between different sample sizes, which is promising in terms of the robustness of the signal that we can get with fMRI drug reactivity. And if we do retesting with the samples that we have shown that, and we know that kind of reliability is a kind of a big challenge in task-based fMRI. And we have shown that in the fMRI drug reactivity, we can have a reasonable replication in terms of the reliability in at least certain areas that we have things over 0.5, I see over 0.5, and in some areas even over 0.7, which is reasonable. Different people try to develop different tasks, and many of these tasks are already publicly available, including the tasks that we designed and we basically provided the resources. And now there are 5-6 other databases that are publicly available for alcohol, cocaine, for cannabis. So people are developing these different databases, and now we have publicly available fMRI drug reactivity tasks that could be used in the field. And the other good thing about fMRI drug reactivity, talking about one of the publications that Anna has made, one of the really kind of interesting works that I really like in Neuron, basically she and Rita Goldstein, they showed that fMRI drug Q-activity could activate all the major neurocircuits that we are interested to target in addiction, which is promising as well in terms of we can engage those networks with fMRI drug Q-activity. And Anna, do you want to kind of mention this slide, or do you want me to just
go ahead? Yeah.
Please, Anna.
This was just as an example of that these neuroimaging biomarkers can also be used to predict clinical outcomes. So we also did a review on which tasks engage networks that predicted later clinical outcomes, and this is an example here for relapse. So the drug reactivity task was one of the best ones for predicting clinical outcomes.
Awesome. And then in terms of the potentials that we have, when we talk about Q-based reactivity, we have a specific paradigms for doing interventions designed for fMRI drug Q-activity. This is one of the paradigms that we designed combining different cognitive interventions, from cognitive memory reconsolidation, episodic foresight. So there are potential for developing specific behavioral interventions. While we know that many of the TMS studies that are happening right now, they are basically Q-based interventions. So even the approval that we already have for deep TMS, for nicotine, and for OCD, they are all Q-based sort of interventions. So they prime people with the drug cues or with the other cues, and then use TMS. So we know that these sort of non-invasive brain stimulations are using Q-based interventions. So this is something like a routine or a kind of a routine practice for developing intervention, which could be helpful for what we are doing for fMRI drug Q-activity as an outcome measure. And then in terms of fMRI drug Q-activity, in terms of the level of evidence that we already have, in terms of the published papers, we have, by the end of 2021, we had 357 studies with 15,000 participants in fMRI drug Q-activity studies. As you can imagine, it's a huge sample size that people recruited for fMRI drug Q-activity studies. And of course, they are being distributed across nicotine, alcohol, cocaine, and across different. I'm sorry, it's by the end of 2020. So by the end of 2020, this is the number of the subjects and participants that we have recruited. And people use fMRI drug Q-activity as an outcome measure for different interventions, behavioral, pharmacological interventions. So it is something like a routine practice in pharmaco-fMRI studies or behavioral fMRI studies to use the fMRI drug Q-activity as an outcome measure in trials. And even in terms of the biomarkers, and based on the definitions that we have for biomarkers, whether they are diagnostic response biomarkers, as Anna mentioned, something like predictive biomarkers, there are reasonable amount of evidence that the people develop over time that fMRI drug Q-activity could be something promising for being used as a biomarker. When we talk about biomarker, one of the main issues for biomarker is biomarker specification, how we are specified that. And we know that with fMRI drug Q-activity, there are lots of methodological details. And we tried together to put together a consensus statement on all the methodological details that an fMRI drug Q-activity should report. This has been recently published in Nature Protocols. And many thanks to all the contributors, and many of them are already online in the meeting that we have. And two brilliant medical students they have, and I really like them to be involved in these processes as the next generation of those people who are interested in this field in the next steps. In that specific methodological protocol and checklist, we developed a kind of list of items. And then we also have explored all the previous publications for a period of time in terms of how much they have been adherent to these items. So we are trying, even right now, we are doing that as something like a live systematic review. So every single new publication that is coming out in fMRI drug Q-activity, we appraise them based on the checklist and kind of define a specific score in terms of how much they have been adherent to the checklist and the items that should be reported in fMRI drug Q-activity studies. So we have discussed about 7 reasons that makes us really excited about fMRI drug Q-activity. But the challenge is that there is no FDA approval so far for fMRI drug reactivity as a biomarker. So that is the main challenge, how we might be able to get that level of quality. So we try to put together a pipeline in collaboration with FDA, with NIDA, with NIAAA, and right now we are discussing that pipeline. That pipeline includes a discovery and definition step, a validation step, and as you can see here, a part of that validation and discussion steps that we have, discovery steps, is what we call basically the mega analysis that we want to do in fMRI drug reactivity kind of consortium within ENIGMA Addiction. So this is a part of the pipeline that we are defining with NIDA and NIAAA, and then we are discussing with FDA to make sure that they would be happy with that pipeline. Right now, we are discussing with them in terms of giving us a letter of support, confirming that they are interested to fMRI drug reactivity as a potential biomarker. And of course, at the end of the day, we are interested to have that as an approved drug development tool, or also in the preclinical use as a biomarker. So we are working on that pipeline, and we hope that ENIGMA Addiction Q reactivity would help us to provide enough evidence for FDA to get to that level of qualification to be able to use this tool. And as the last slide, one of the things that people might think is that, I mean, we have a large level of diversity in terms of the methodological parameter space for fMRI drug reactivity people have done so far. But the good thing is that there are certain kind of number of studies that they use very similar methodological structure. For example, we have 252 studies that they have used pictures, but we have 217 studies that they have block design. So that makes it easier for us to be able to do some collective effort on that line. We have already kind of started to put together a list of labs that are interested to share their databases. Right now, we have more than 1,000 data points for patients with fMRI drug reactivity, which is promising. And there are many other people who are already online who are interested to contribute to this database. So we are working to be able to make that happen. And thank you very much for your attention. And I hope that with support that we have from you, from Patricia, from Scott, and as you can imagine, they have a great colleague like Anna, we will be able to make that happen together, and we will see the results in the next coming years, hopefully.